Okay, it looks like we have a good amount of participants on today. So good afternoon, and thank you for joining us for today's webinar, Innovations in Biosecurity Measures for Feed Mills. I'm your moderator, Elise Schaefer, publisher of Feed and Grain magazine. And our speaker today is Dr. Cassie Jones, associate professor at Kansas State University. Over the next half hour, she'll share her highlights of what she's learned about feed mill biosecurity by studying the spread of African swine fever. So once again, I want to thank you for spending part of your very busy day with us, and I especially want to thank our sponsor, WEM Automation. WEM Automation is an American-owned high-precision automation and process control systems company that provides animal nutrition manufacturing solutions from New Berlin, Wisconsin. Since 1955, WEM has provided flexible solutions that fit your process to optimize your performance. Their industry knowledge and 65 plus years of experience with feed milling, grain storage, and premix facilities enables them to provide effective solutions and increase profitability for the smallest of processes to the largest full facility control systems. The WEM industry leading technical service group is available 24/7 and can access your system remotely from New Berlin, Wisconsin. So thank you again to WEM. And with that, I'll turn things over to Cassie.
All right, thank you so much. And thank you to both WEM and Feed and Grain for the invitation to visit with everybody today. I'm excited to get to visit with this group. I have, as I'm going through the participants list, I see lots of familiar names, people that I've been able to visit with previously at different conferences, and I miss you all. And so I'm excited that we have something like Feed and Grain Live where we can still share some of our research and share some of our updates with you, but am very much anticipating getting back into the conference type setting where we can have more open dialogue. So please, as they stated, feel free to be sending questions or comments in the chat, and we'll have time at the end for all of those. Today, I was asked to talk about some of the innovations in biosecurity measures for feed mills and just give an update on where we are from a lot of our research. And I actually have brand new data that I'm excited to share with you today as well. So to start with, this is very much a presentation in my research area is focused on the control of biological hazards. And this is a term that we've probably become too familiar with over the past year and really over the past decade with FSMA. But there are certain types of biological hazards that have the potential to be carried by feed. Historically, we've thought about those like prions, and that was maybe one of the first major changes to our food safety and feed safety rules was with bovine spongiform encephalopathy transmission through the feed supply chain. We also know, because in some of our industries, it's more important than in others, that salmonella has the potential to be transmitted through something like a feeder ingredient, as well as listeria. About 5 to 6 years ago, we started to recognize that viruses could also be transmitted through a feed or ingredient. And we initially saw that with porcine epidemic diarrhea virus. And now, of course, we're all on alert about African swine fever virus as we see it really ravaging a number of our important trade partners. But I do want to make sure that we're very clear that it's not like these viruses or pathogens or biological hazards just magically jump from one place to another, that we have very clear methods of disease transmission. And that takes something where we initially have both a reservoir and a host. It's not like we magically have feed that gets contaminated with ASF. We first need to have a reservoir, a habitat where that agent lives, grows, or multiplies. And we also have to have a host, an individual that's susceptible to that specific agent. And so if both of those are true, then we have the potential to transmit disease from the reservoir to the host. And it's most likely to occur through some type of direct transport, whether that's nose-to-nose contact of pigs, or like we're seeing with COVID right now, direct contact by individuals with others as you're getting sneezed on in an airplane, or are sitting in a classroom or in an office setting with someone who's been infected. So we know that direct transmission is by far the most likely method of disease transmission. And that is absolutely true for all of the viruses, all of the biological hazards that I mentioned previously. However, those are the ones that we've really done a pretty good job in the feed industry, in the animal production industry of controlling. And so that has opened up the opportunity for indirect transmission to play a larger role. Again, the most likely method of transmission is always going to be direct. But once we take care of that hurdle or fill in that gap, it does leave some gaps or opportunities for indirect transmission. That's where we're thinking about indirect transmission. This is maybe sitting 30 or 40 feet away from that person that would be COVID positive. Or that's really the role that feed or ingredients or biosecurity or lack of biosecurity potentially could play in the transmission of some type of disease from a reservoir to a susceptible host. And what I want to share with you today is some of our newest data for how much of an issue this might be with a specific virus of concern. And of course, the one that we're all talking about recently would be African swine fever virus. We've done a lot of work in both salmonella as well as porcine epidemic diarrhea virus to try to understand the potential for indirect transmission. But this is some of the first work that's been done in this case with ASF. We know that ASF survives for a really long time. We know that if we feed contaminated feed to a pig, that they can get sick. But what we don't always know is how easy it is for that indirect transmission to occur. And so that's really what the focus of this study was, which is how feed can serve as a vector of disease, and specifically in this case of African swine fever virus. Now, this was done, this type of research was done at our Biosecurity Research Institute, which is a biosafety level 3 facility, meaning that we have to have FBI clearance to have access to the facility. It's obviously gated, and we're working directly with African swine fever virus. We have this facility here in Manhattan, Kansas. The other major facility for which we do this work in the United States would be Plum Island currently. And so we're fortunate that we have the ability to access it, but I do want to emphasize that this was done under very much controlled conditions. So what we did is we wanted to initially evaluate the impact of African swine fever virus contaminated ingredients on the contamination of subsequent batches of feed manufactured in the same equipment. And the thought process here is that you all in the feed and ingredient industry have demonstrated very clearly in a variety of different hazards, whether they're chemical in nature, like some type of drug carryover or nutritional toxicity concern, or even biological in nature, that sequencing and flushing is really one of the easiest, most practical ways to get rid of some type of contamination. And so we wanted to look at, well, if we have a contaminated ingredient that happens to come into our mill or into our production, into our system, where and how much does that contamination spread? And how many kind of batches of feed could it potentially impact? And so that's what we did here. So we first started with a negative batch of feed that we manufactured. So within this biosafety level 3 facility, when we're working in full containment, we built a feed mill with the help of Charles Stark to make feed and contaminated feed in this type of environment. And so initially we made this batch of feed and we first primed the system with negative ingredients. And so we put all of the ingredients into the mixer and then we conveyed them through a bucket elevator. And then this is where things started to get a little different, is that then we discharged them into a biohazard tote or a biohazard bag instead of into a truck or into 50-pound bags or 1-ton tote bags. And so this is where we are working with pilot scale equipment, but still this is the equipment that we use for testing phytase stability and drug carryover in our different feed mill production settings. And so then after we collected or manufactured that first batch of feed, all with negative ingredients, then we took samples from within that tote from 10 different locations and analyzed those samples for African swine fever virus. So we did that initially with one batch, all of which those ingredients were ASF negative. That was our control. And then we manufactured a batch of feed that contained an ingredient that we had spiked with African swine fever virus. And so we manufactured again, put ingredients in, but one of those ingredients was ASF positive. We conveyed it, we discharged it, we sampled it. But then we manufactured another batch of feed because our real goal here was what happens if we have a pulse of contamination that comes into the mill. And so we manufactured another batch of feed, all with negative ingredients, and actually 4 subsequent batches of feed, really trying to test the premise of how long does it take for us to flush out any of the contamination from the manufacturing equipment, and how likely are we to detect ASF in any of these feed samples that are produced 1, 2, 3, or 4 sequences after we manufacture that contaminated batch. So here's the data. So I want to set it up first. So this is just in table form because it's preliminary data. We're just getting the results back on these. And so we have the different batches of feed. So I'm going to show you batch 1 first. That was our batch with negative ingredients. And as you would expect, when we analyzed the 10 feed samples, that's why feed is underlined because we were analyzing feed here. When we analyzed the 10 feed samples, none of those had detectable levels of African swine fever virus, which is exactly what we would expect, right? Because this is before we even entered ASF into the manufacturing system. None had suspect levels, none had positive. So all of them had non-detectable levels of African swine fever virus. Again, as you might expect, after the positive batch of feed, we had all of our 10 samples had African swine fever virus. So this again is probably the 4th major research study that we've demonstrated that we can mix viruses into feed just like any other ingredient that we add in. And so if we have contamination hit a mill and then we have something to homogenize those ingredients like a mixer, that mixer is not only making sure that we're distributing our enzymes and our medications and our vitamins very clearly or very consistently, it's also very evenly distributing African swine fever virus. And so all 10 of the feed samples that we collected were ASF positive. What we were more interested in, though, was what happens next. So how much kind of carryover was there into the next batches? So remember, these next subsequent batches did not contain African swine fever virus contaminated ingredients. They just, if there was any contamination, it would be from residual contamination from that second batch. And what we found is that in the batch immediately following, and the batch after that, and the batch after that, and the batch after that, all of them had some level of either positive or at least suspect levels of African swine fever virus contamination. None of those samples, as we look at that very first row, had anything that we would consider to be non-detectable. When we say suspect, that means that it's in this kind of gray area where the PCR computer is telling us we think that there's something here, but we can't amplify it high enough. And so it's a suspect level. But 7 of the feed samples, even 4 sequences later of negative ingredients, we're still finding ASF presence within those batches, showing that this risk really does exist. Now, a really relevant question next would be, what are the levels though? And so what we would expect to find here is decreasing levels of African swine fever virus. It's concerning that we're detecting it, but we are reducing the concentration as we continue to make negative batches. So when we initially manufactured that positive batch, it had a CT of 33. And CT is represented in cycle threshold. So that's really how many times the machine needs to amplify the sample before it can detect. So a lower number is typically equivalent to more African swine fever virus. Alternatively, another way to look at this is looking at the number of genomic copies, and this is on a log basis. This is maybe people who are more comfortable looking at something like salmonella. This is a number that might be more familiar to you. So like 4 logs versus 3 logs versus 2 logs of virus in terms of genomic copies. Regardless of the way you look at it, the numbers and the quantity of African swine fever virus is reduced, but not as much as we would hope. We only saw a 2-log reduction from our positive batch. We still had relatively high levels of African swine fever virus that were detected within the manufacturing equipment 4 sequences later. Remember, all of those ingredients were initially negative. So I want to also zero in just on this positive batch, because then we did some additional testing. So we knew that this one would be positive, and so we took these samples and we wanted to evaluate how long they might be positive and how long they're going to maintain this virus. Well, if you know much about African swine fever virus, you know it's really durable, but this really is where this research differs pretty dramatically from things that we've seen with both salmonella and something like porcine epidemic diarrhea virus. Because as we look at that level of virus, whether it's in the quantity of ASF detected in genomic copies, so this is around that 4 log from day 0 all the way out to day 180, you can see we have a really stable virus within feed. If we look at another measure, this is in CT, again, really stable from day 0 all the way out through day 180, we have really substantial stability of African swine fever virus. And so if it makes its way into a feed mill, or into the feed supply chain, it can stick around for a long time and can impact lots of subsequent batches. The last part of this research study was really focused on the equipment and environment, because we do know that we can impact risk and kind of sequence ourselves away from risk at times. But one of our challenges is always what happens to the manufacturing environment, and is there risk, especially when we talk about something like biosecurity, in terms of truck traffic and personnel traffic from the mill to farms and back to the mill. And so in this case, we, in the same type of system, evaluated what's happening to the equipment and the environment. And so to do this, we have our main parts of our feed mill that we set up in containment, and any feed contact surfaces like the inside of the mixers, conveyors, or bucket elevator were considered to be zone A. And then we sampled within 1 meter of those manufacturing surfaces, but not in direct contact. So this is like the floors and walls and the outside of the equipment that's nearby. That would be considered to be zone B. And then we have the surfaces that were much further away. So floors, walls, ceilings that were more than 1 meter away were considered to be zone C. Now, you all know in the feed and grain industry that those zones are pretty straightforward, but then we have people and things and vehicles moving in and out of those zones all the time, especially between zones B and C. And so we had, in this case, a zone D, which was represented by the clothing and the shoes of our researchers that were working, because they were constantly moving in and out between zone B and C. We would refer to that as a transient zone, because it transferred or went back and forth between those zones. So what I have next is the prevalence of African swine fever virus on those surfaces. So whether that would be a feed contact surface, surfaces that are nearby, surfaces that are further away, or these transient surfaces. And then along the x-axis down here, I have when we collected those samples. So we have the negative versus the positive after that first batch of feed that was manufactured that was ASF positive, and then the 4 negative sequences afterward. So first we'll look at the feed contact surfaces. And as you can imagine, after the first batch of feed was manufactured with ASF negative ingredients, there was no ASF detected on the equipment as we would expect. As soon as we introduced ASF as an ingredient into the manufacturing equipment, the environment and equipment samples collected afterward on the feed contact surfaces, the inside of the mixer, the inside of the conveyor, pretty much all the feed dust that was left, had about 75% of those surfaces were still ASF positive. And we had kind of bouncing levels, but still about on average, half of those surfaces remained contaminated with ASF on the inside of the mixer and the inside of the bucket elevator, et cetera. And that really, again, reinforces what we see in the feed samples, that this level of contamination continues to exist within the equipment long after we had introduced and realistically gotten rid of the contaminated ingredients. Now let's look at what happens outside of those manufacturing surfaces. So the nearby surfaces and those surfaces that are further away also get contaminated. And so this poses risk as we think about this contamination event initially occurring and then someone or something landing there and potentially carrying it to a different location for transmission to occur. The one that's perhaps most concerning would be the transient surfaces that are constantly in and out of between these zones And those surfaces went positive as soon as, right away after we manufactured the positive batch of feed, and they remained positive. This is something we've seen now. This is the third research study where the people that are manufacturing the feed are more contaminated than the feed manufacturing surfaces themselves. And so I think it's really important for us to consider when we evaluate biosecurity, when we think about it in a holistic sense, is making sure that we're accounting not just for the ingredients, but also for the people and the vehicles that are part of that feed manufacturing process. And so that's because as we think about potential transmission in the feed supply chain, right now we're talking about African swine fever virus. And if that's the case, as we sit here today in March of 2021, we are still ASF negative in the United States. And both fortunate to be so, but also a credit to you within the feed supply chain, a credit to our pork producers that we have continued to make this a priority and have been able to maintain our ASF-free negative status or ASF negative status. But we anticipate that if there's risk here in the feed supply chain, this is what would happen, is that we think if it's going to enter, it would come in through an ingredient. Again, this would be if it's entering as a foreign animal disease. From an ingredient, then, we're concerned that it might get distributed to a feed mill, and from that feed mill through trucks to different farms, and ultimately cause infection within an animal. However, what we've seen in other cases and in other viruses domestically, as well as with African swine fever virus in Vietnam and a lot of our work there, is that the feed supply chain can still be implicated even if it wasn't initially in an ingredient. And this is why biosecurity becomes really important, is because as we think about an infected animal at an infected farm, it doesn't take long for that entire farm to be contaminated. At that point, though, we're moving a feed truck and people back and forth between that farm and different feed mills. Now, we have a lot of things in place to prevent farm-to-farm transmission, but every day we have feed trucks going from farms to mills to other farms with, in my opinion right now, little to no biosecurity, at least in a biological sense. That's very different from what we're seeing in a country like Vietnam, where we have ASF endemically involved. And so in the worst case scenario, what could happen is a feed truck can pick up this contamination on a farm and become contaminated, whether it's on the truck tires or in the inside of the cab. That can be transmitted to the mill, in which we can then contaminate other farms, potentially. Ingredient trucks headed back to the ingredient facility can carry it, and we can have kind of blow-ups across other production systems through what I would consider to be backward transmission through the feed supply chain. And so I bring this up because I want to just emphasize that as an animal food industry, we have a responsibility. So to start with, I want to really emphasize animal food is not the most likely way that a foreign animal disease like African swine fever virus or anything else is going to enter the United States, in my opinion. I don't think that it's the most likely way. We know direct nose-to-nose contact, direct transmission is much more common, much more likely. We know that there are much bigger gaps that we need to identify or that we need to focus on, but we know it's possible. And so we need to take care of our business. This is our business and this is our segment. And so we have an obligation to recognize the science here supports that transmission can occur. And historically, we have seen the feed supply chain be a source of foreign animal disease entry into otherwise naive countries more than once. So as we look at porcine epidemic diarrhea virus, USDA has come back and said, we still don't know how it came into the US, but the most likely source were tote bags from China, not the ingredients themselves, but that the tote bags that were carrying those ingredients were reused and had some level of PEDD entry. Alternatively, we know that it's been documented that foot and mouth disease has entered naive countries, both Japan and South Korea, through a specific ingredient, whether that's rice straw from China or forage and hay from somewhere in Southeast Asia. And that's been confirmed by both the Japanese ministry and the South Korean Animal and Plant Quarantine Agency. And so we know that it can occur. We know foreign animal disease can enter through a feed or ingredient. It's not the most likely way, but it can occur. And so the challenge, especially within our feed supply chain, is that if pathogens were to enter, whether it's through a tote bag or through a contaminated ingredient, or even if this is an endemic pathogen that we already have here, like PRRS, but it gets back onto a feed truck and then back into a mill, we can very quickly spread that virus or spread that pathogen all over because of the way that our feed supply chain is set up and so intricately linked to multiple farms. So what can we do? What can we do to limit pathogen entry or transmission through this feed supply chain? So there are kind of three things that have to perfectly align in order for something like this to actually occur. First, we have to have an ingredient get contaminated with some type of pathogen of concern, like African swine fever virus, and for that that ingredient to be destined for United States pigs. Second, even if it ends up in product X from country Y and is intended to come into the United States, it has to survive that transport, which takes anywhere from 3 to 6 months. And then finally, it has to come in at a level and in a form that's capable of causing infection. And so ultimately, what are we doing with biosecurity right now? We're trying to address all three of them because we don't know when any of these things have occurred. And so the best strategy when we don't know which one is most likely to occur is we try to slay all three dragons simultaneously. So the first component of this is to exclude high-risk ingredients altogether. This is something that actually I've been on lots of webinars, lots of conference calls lately as we're trying to figure out how to go about this in the most strategic sense. But more than anything, know your supplier and their supplier and their supplier. And so if you're concerned about an ingredient and you say or make statements like, oh, we don't have anything from an ASF endemic region, but do you? So this is who you purchased that product from and it has their tag on it, but where did they get that initial product? And so really knowing your supply chain is important. Requiring biosecurity during manufacture, storage, and transport and demonstration of that prior to its arrival at your own facility. And in my opinion, excluding high-risk ingredients altogether. There are segments of our industry that need to and rely heavily upon specific types of ingredients like organic soybean meal, but we don't need to be pulling in soybean meal from ASF endemic countries, in my opinion. Those are practices that I think are just too dangerous based on the data. And so completely excluding those ingredients from our mills altogether, I think is very important. The second step here would be to take advantage of opportunities to reduce the survival of infectious virus and contaminated ingredients. Some viruses are more likely to survive than others. What we know about African swine fever virus, though, is that, man, it can survive for a really long time. And so looking at from the point of manufacture to when it possibly could have gotten contaminated until you actually use that product is an important part of this. And then really what we want to kind of focus on in the last minute or so here is preventing the potential for farm to mill to farm contamination. And that's where we really look at feed mill biosecurity. There's a lot of farm to farm biosecurity measures in terms of controlling foot traffic, including controlling vehicle traffic, controlling personnel that are in and out of the mill or facility, controlling shoes, and having people change shoes that are the norm within animal production facilities today. That's not the case with feed mills. And so one of the things that I find to be most helpful is to use a product called Glow Germ. And this is really intended for like an elementary school human pathogen safety, like a hand washing experiment. But what we've been doing is using it to evaluate biosecurity compliance and gaps. And so we'll put Glow Germ all over the floor of our feed mill. It's something that when it's just looked at in normal light, it's kind of a white powder that you might see, but usually it's kind of invisible, but it fluoresces under UV light. And so you can see then, and this is a Danish bench into a pig farm, that over time you get a lot of tracking of that powder, and really it's an indicator for potential risk into a certain point of your farm. But then we need to put things in place, like in this case, a bench where people take off their shoes and then they come over, and on this side of the bench, you don't see any of that powder, showing that you have stopped the transmission or prevented that transmission from coming through. Looking at something like this within a feed mill has become really helpful for us, trying to identify in a case-by-case scenario how to identify where our biggest gaps are and where we have foot traffic that we need to be tightening up. So I just wanted to kind of conclude with what does feed biosecurity look like today? It depends. It depends on the production system, it depends on the mill, it depends on the ingredient, but this is everything that I can see from across the world. And so we have some mills in the United States that are changing shoes. And sorry, my phone's ringing. And we have Danish benches, or people are using quite a bit of floor powders and dry sanitizers. We're having people in some facilities, ingredient facilities and mills, putting on booties or different suits over their clothing. Some places in the United States, we're starting to see ingredient truck and feed truck washes, similar to what we would with live haul trailers. We see that more commonly in areas with foreign animal diseases, like this is a picture from our partners in Vietnam. And then looking at different innovations in terms of ingredient pit covers, how we're backing up to ingredient pits instead of driving over them, some steam processing options. And so these are all things that we're working on currently to evaluate feed biosecurity. We have a lot of different projects right now. So right now, one of our projects is looking at showering versus some of these benches versus foot baths on preventing entry of contaminants into a mill or into a facility. We're looking at how to decontaminate the inside of feed truck cabs, because it's one thing where we can sanitize the outside of them in high risk times, but one of our biggest challenges in Vietnam currently with our partners is that the truck driver gets out from at one site, goes up, climbs up a bin and opens the bin lid so that they can put feed in, and the bottom of his shoes and his clothing get contaminated, and then the inside of his truck cab becomes contaminated. We're looking at development of an in vitro model to evaluate the infectivity of swine viruses in feed or ingredients, trying to quantify some soy imports into the United States from ASF endemic regions, and then facilitating the development of a proposal for a gateway program for importing some of these non-animal origin feed and ingredients from ASF endemic regions. So if we're going to be bringing in some of these because we have to or can't source them economically from other locations, what should we be doing, and what types of paperwork or processes should come with these ingredients to make sure that they can be used safely? So to finish up, the feed supply chain, again, is not the most likely route of viral entry into animals, but it can quickly spread disease. Because of that, we all play a role in preventing pathogen entry and spread. So I just ask that we all do our part. For me, that means excluding high-risk ingredients, not just from my formulas, but from the mill or the facility altogether. It means consider some active mitigation to further minimize risk of disease for survival and infectivity, and implementing a lot of this feed mill biosecurity. Finally, I ask that you continue to help us understand the problem and potential solutions. Our best work has been done where we've collaborated with industry on real world problems. And so it's really when we hear from you saying, this is our biggest challenge right now. We want to do this, but this is our barrier. And helping to resolve those so that we can implement this on a practical basis is really where we like to be at that center point of work. So my email is listed here below. You're welcome to email me if you have any questions, but also I'll open up at this time to any questions that you might have.
Great. Thank you so much, Cassie. That was an enlightening presentation, and really appreciate you sharing what you learned about African swine fever and applying that to biosecurity in general. So folks, just a reminder that you can enter your questions with the chat window. And I do have a few more, or a few that came in, but feel free to enter them now if you've been hanging on to it. So Cassie, I'll start out with, how would the method of preparing feed make a difference in biosecurity? For example, would pelleting the feed help versus extruding?
Yeah, that's a great question. And we have not ever tested that in something like African swine fever virus, but we certainly have with PED. And then we've modeled it with ASF. And so what I'll tell you is with something like porcine epidemic diarrhea virus, we know that thermal processing, whether it's pelleting or extrusion, will kill it. But it does not prevent post-processing cross-contamination. And so when we are pelleting something, but our immediate next step is to put it into a cooler, and we're pulling air in, ambient air from the facility itself, that air is not necessarily sanitary, right? And so we see that pelleting, at least with PEDV, a lot of our early data demonstrated that pelleting reduced the concentration of the pathogen, because we kind of went from like a 4-log to a 2-log, but it didn't completely remove the contamination altogether, because it did it first, but then we just cross-contaminated it or recontaminated it during the cooling and distribution process, because it's not like, you know, as soon as we introduce that pathogen into the air, it seems to kind of go everywhere within a mill. Now, again, we don't know that in ASF. We have not done the thermal processing studies. We've modeled them, and theoretically, pelleting and temperatures typical of pelleting should destroy African swine fever virus. But again, that's if 100% of your product is reaching temperature, which the beginning of a run wouldn't. And if you have any plugs, it wouldn't. And then you have complete control or prevention of recontamination after the point of that thermal processing from occurring. And again, typically we don't. So what we typically see in a realistic environment, and again, I'm pulling on my experience from Vietnam here, is that we would have pelleting occur, and if there was contamination in that initial ingredient, it would kill it, but then we would blow it back on during the drying process, and certainly during the transport and distribution process onto farms.
Okay, great. Thank you, Cassie. I've got another question. What would you classify as a high-risk ingredient?
Yeah, that's a great question, because it's different for everybody. For me, high-risk ingredients are grains, oil seeds, or really anything that could potentially be unprocessed from an ASF endemic region. That'd be one category of them. And so I just served on an EFSA panel, a European Food Safety Authority panel, where we were trying to characterize these. And it astounded me how, you know, their world is feral pig populations and wild boar populations, and where they're struggling with ASF would be these wild boar who are dying in cornfields. Or because as you have a combine going through the field, then picking up that carcass and having contamination at that point during harvest, where it's not as much of an issue in something like small grains, or even sometimes in soybeans, because you can typically see those carcasses and you can kind of drive around them. But corn was one that they were more worried about because of the potential contamination in terms of the carcass of dead animals, which I had never really thought about. But as you think through that, we need just the potential to be contaminated in the first place. And so that's where I come up with grains and oil seeds from regions where we know that we have endemic disease. But then once we look at kind of that next step, I don't think that that vitamins and all enzymes or amino acids, those probably aren't going to be considered to be as high risk, in my opinion, even if they're from the same region, because there's just not the likelihood that they would get contaminated. And so there's a lot of things we can do, but those for sure would be the top of my list for high risk ingredients, would be grains and oil seeds from endemic regions. Then after that, I think that we have all other ingredients from endemic regions, just because of the potential for cross-contamination during transport. So it's, again, I'm probably not worried about the stuff that's inside the bag or the tote as much as I am the outside of the bag or the tote itself. And then after that, I do worry at times about porcine-based ingredients not being the source of entry into the United States for something like a foreign animal disease, but being a potential route of rapid spread. And so because of that, we have a lot of producers that just choose to not go that direction.
Okay, great. Thank you for breaking that down for us into different categories of risk. I have seen, Cassie, several questions about the details of the study, such as the variables, where were the ingredients coming from that were in your feed samples. Is there somewhere online people can go and look at those details a little closer?
Yeah, great question. Not yet, because I'm telling you, this is like brand new data. So you'll see things from researchers in different ways. K-State tends to, and our team tends to kind of publish or release things as soon as we have that information, because we think, especially in something like this, it's so important to get you the information. We don't have that published yet. And so we're actively working on the statistical analysis on it, writing up all of the paper. It's going to be another 6 months before the paper gets through the review process and is accepted, all of those things. And we think that it's so important to get this information pushed out to you now. And so we don't have all of those details in some place, but I'm happy to share them with you. This is a National Pork Board funded project. And so, to be honest, the ingredients themselves aren't as important. And so we introduced a contaminant and we just spiked a batch of the feed. So it's a typical corn, soybean, meal-based diet that would be very similar in formulation to either a phase 3 nursery or to a sow gestation diet. Both of them in our world is very similar. So there's no fish meal. I saw that question. There's no medications. There's no preventatives that we added. We just wanted to look at kind of a typical corn, soy-based diet that would be very representative of what we feed here in the United States to quite a few pigs. And then we took a part of that diet, took it and contaminated just that kind of 1 pound, and then added that back into the mixer. And if I remember right, this was a 50 kilogram mixer. And so certainly not super large scale, but you can imagine our constraints from doing this in a biosecure facility. And so we did the best we could with having pilot scale but representative equipment. And so we confirmed that like the mixer was giving us a similar CV and had a mixing time, a dry mix time associated with it, that we would actually the same specs that we would use for like phytase or feed additive studies. Same with the bucket elevator, as we confirmed it was just a smaller scaled down version. The major change was distributing into biohazard totes instead of into a feed truck or into a typical 1-ton tote.
Oh, and sorry about that, folks. Looks like I was on mute, but thank you, Cassie. And at this time, this is all the time we will have for questions, but Cassie has her email address there at the end of her presentation. You will all be getting a copy of this with her email address included, as well as a full recording of today's webinar. And so I just want to thank you very much, Cassie, for your time. I want to thank everyone who signed on with us today, and I especially want to thank our sponsor, WEM. And I would welcome you all to come to our website, live.feedandgrain.com, to see upcoming webinars as well as today's recording. Thanks, everybody. Bye-bye.